Okay, so we are going to finish by talking about hemoglobin and myoglobin function and our oxygen saturation curves. Okay, so we are going to be looking at a pretty cool case, and I'm going to zoom in a little bit so we can see the thing bigger. And so this is kind of like the Jurassic Park of biochemistry questions, but not with something from the Ice Age rather than the age of the dinosaurs. So a group of researchers actually were able to extract DNA from a Siberian woolly mammoth, and they were able to locate the sequence of the hemoglobin gene. So they cloned that into an organism and produced woolly mammoth hemoglobin. And so they looked at the properties and compared it to a very similar animal, genetically speaking, the Asian elephant, to see, why could woolly mammoths, was their hemoglobin different in its properties that adapted it to living in colder temperatures than the Asian elephant, which is a modern animal. So they generated oxygen binding curves for both forms, the woolly mammoth and the Asian elephant. And so we're first going to look at the solid lines, and we want to look at affinity. Okay, so when we're looking at oxygen saturation curves with hemoglobin or myoglobin, okay, we're going to look in terms of affinity, we're going to compare P50 values. Okay. All right. So if we compare the P50 values, right, I'm going to change this to a dashed line. Okay, we're going to figure out what those P50 values are. Okay, by, again, looking what is our oxygen pressure, okay, at which we get 50% binding. Okay, so what we want is, first we're going to look at the woolly mammoth, so we'll do that in blue. And when we go on the x-axis to find out what the partial pressure of oxygen is, we find that the woolly mammoth is 246. Okay, so mammoth P50 is about 6 millimolars of millimeters of mercury. This is a pressure. Right now we're going to look at the Asian elephant, and when we drop the line from the 50 onto the x-axis, we get about 12 millimeters of mercury. This is the Asian elephant. Okay, so we want a higher affinity. So higher affinity is going to correlate with a lower P50. Okay, lower P50 means you don't need as much oxygen around to get, half of your sites bound. So means higher affinity. Okay, so the other thing is, remember when our curve is further to the left, that corresponds with a lower P50 and a higher affinity. So curve more to left on graph, is lower P50 and higher affinity. So if we're talking about who wins the affinity for oxygen competition, that is going to be the woolly mammoth. Okay, so now if we want to think about why this might be, why would it be to the advantage of the woolly mammoth to have its hemoglobin have a higher affinity for oxygen? Think about what you may have studied in Gen Chem about chemical reactions and temperature. So this is a chemical reaction that's just binding, right? So if we're just thinking about this as a binding reaction, we've got our hemoglobin plus our oxygen is making our hemoglobin bound to oxygen. Okay. What happens when chemical reactions are cooled down? Or what happens when chemical reactions are sped up, right? So temperature has an effect on chemical reactions, right? Chemical reactions are faster. They're usually faster at higher temperatures. So if the woolly mammoth is living in a colder temperature, okay, then this chemical reaction, the binding reaction, the molecules will be moving slower and they'll be encountering each other less frequently. And so if the woolly mammoth didn't have a higher affinity for oxygen, it would be, at those cooler temperatures, it's going to be less efficient at binding oxygen. Okay, so having a higher affinity for the oxygen is beneficial when you're living at colder temperatures. So that's great. Okay, so now let's consider these dashed lines, which are different conditions. So they represent when we have these two molecules, chloride ions and this molecule BPG, that after break we'll be talking about its effect on hemoglobin. Okay, so but for this case, all we want to look at is what happens under these conditions to the affinity for hemoglobin. So we're going to, just because we like to drop lines and figure out P50s, let's think about this. Okay, so first thing, it appears to me that the two lines almost directly superimpose. Okay, so under these conditions, whoops, let's change that back to a solid line. Okay, so under these conditions, there's little to no difference between the two species. So that's the first observation we can make. Okay, the second is we're going to drop this line and figure out, what our P50 value is. And so our chloride ion plus BPG conditions, the P50 is equal to what? So let's see, 12, 14, 16, let's say 17 millimeters of mercury. Okay, so it has a larger P50. The curve is shifted to the right. So what that means is that under these conditions, okay, there is a lower affinity for oxygen. And again, after break, we'll talk about BPG in humans, not just woolly mammoths, and we'll see that this is a molecule that helps hemoglobin to unload oxygen, because you need to unload oxygen as well as to load it, right, for it to function. It needs to pick it up in the lungs and deliver it in the tissues. And so if you're woolly mammoth and you have the ability to bind to oxygen more strongly, then that might cause a problem for you when you actually need to use the oxygen and deliver it. So having, these effectors, these molecules that can shift the behavior of your hemoglobin and make it have less of an affinity, can help you with the delivery. So if you can control it so that you don't have those things when you're picking up in the lungs, but in the tissues you do have these molecules, it makes it easier to deliver. All right, so that's the story of the woolly mammoth. And so now we're going to talk about hemoglobin and myoglobin, just thinking about, what's true. So we have a statement about myoglobin where it transports oxygen while hemoglobin stores it. Myoglobin has quaternary structure, but hemoglobin does not. Myoglobin displays hyperbolic binding while hemoglobin displays cooperativity. Myoglobin binds iron while hemoglobin binds heme. All right, so let's go and we'll eliminate the ones that we know are false, okay? All right, so we're going to put red X's on them. All right, first we'll start with A. Myoglobin transports oxygen while hemoglobin stores it. Well, that's exactly the reverse of what we know, right? Hemoglobin is the transport and myoglobin is the storage, right? Myoglobin is for storage. So that one is not the answer. Myoglobin has quaternary structure, but hemoglobin does not, right? Well, that's also exactly the reverse, right? So that's wrong. So hemoglobin has 4 subunits, and we talked about this in class, which is quaternary structure. Myoglobin, only 1 chain. So it doesn't have quaternary structure. Okay, so in terms of the next one, oops, what did I do there? Myoglobin displays hyperbolic binding while hemoglobin displays cooperativity. We haven't in class gotten a chance to talk about the hemoglobin cooperativity too much, okay? But we did talk about I did ask you about the shape of the curves. So for this one, when we talk about hyperbolic versus sigmoidal, we've got this shape, which is hyperbolic. And that's what the myoglobin curve looks like. And we have kind of an S shape, okay, which we call sigmoidal or S-shaped. And that's hemoglobin. So the first part of that is true. We'll hold off on the second and see if we can eliminate the fourth option in here. Okay, so myoglobin binds iron 2, while hemoglobin binds heme, right? What do we know about myoglobin and hemoglobin? They have, one hemoglobin subunit looks exactly in its overall structure like a myoglobin. So, but we know that both molecules bind heme, and the heme has iron in it, right? So we can't say that only one binds to heme, and we can't say that only one binds to iron. So that's false. So that leaves us with this one. So even if you didn't remember or know or we hadn't covered hemoglobin having cooperativity, you could eliminate all the other options for this one. Okay, I'm going to end the hemoglobin video there, and